Like we have seen in the earlier chapter, earlier topics we had seen that I had covered female gametophyte, and apart from female gametophyte, endosperm was also dealt with the four types of endosperm, of which ruminant endosperm was not of that importance. That's why I emphasized on the three endosperms: nuclear, cellular and halobial endosperm. And now I went for endosperm because it is on the endosperm only that the embryo will develop. Now, prior to developing of the embryo, shall we go again to the female gametophyte? We'll see the female gametophyte again. Like we had done it earlier, just for this embryo, we have to do it again. This is the embryo sac, and we knew that here were the polar nuclei, here the egg and the synergids, here the antipodal cells. I'll mark it again. Antipodal cells, egg, synergids. Just below the synergids, there are some cell thickenings. Cells are slightly thickened. So these thickenings are called as filiform apparatus. These thickenings are called as filiform apparatus. It is the micropylar end, micropyle. This is the stalk or funiculus, stalk, or it can also be called as funiculus. The two integuments, as I said, based on the number of integuments, it can be unitegmic or bitegmic. Unitegmic means one integument, bitegmic means two integuments. Then these two polar nuclei. This is the chalazial end. What we had seen, the polar grain, the two male nuclei along with the pollen tube, they reach the egg through the micropylar end. Through the micropylar end, they will reach the egg. What is the function of the filiform apparatus? The filiform apparatus also have got some chemical substances. They are chemitropic because due to the chemicals, they are directing the male nuclei towards the egg so that fertilization should take place. That is the function of filiform apparatus. And they are nutritive also. Along with the synergids, synergids are also nutritive. They get the nutrition from other parts of the ovary or the nucellus. From this region also they will get the nutrition for the egg. When it will get developed into the zygote, they will help in that. So we had seen when fertilization takes place, the two male nuclei, of which one will fuse with the egg, the pollen grains, two nuclei, male nuclei. One male nuclei will fuse with the egg and it will form the zygote. Other male nuclei will fuse with the polar nuclei and it will form the endosperm. In the earlier chapter, in this chapter itself, in the earlier topic, we had seen how the endosperm is being developing. And now, so this type of fertilization, see, twice it has formed. Once it has fused with the egg and it has formed zygote. Other time it has fertilized with the polar nuclear and it has formed endosperm. This topic I have dealt with thrice. So you can imagine the importance of this topic, female gametophyte. That is double fertilization. Thrice I have mentioned this double fertilization. Twice it will fertilize, once with the egg and another time with the polar nuclei. That is known as double fertilization, or it can also be known as triple fusion. It can be also be known as triple fusion. Why it is known as triple fusion? Because three nuclei are involved in this. 1, 2, 3. One nuclei of the egg and two nuclei of the polar nuclei, two nuclei. That is three nuclei. Three haploid nuclei are fused, not together, separately. One with the male nuclei and another two with another male nuclei. It is because the fusion of three separate male nuclei are taking place. So then it is known as triple fusion. One male nuclei, one haploid nuclei is your egg cell, and 2 haploid nuclei are of your polar nuclei. So hence it is known as triple fusion. Now, so this is the embryo sac. This is the embryo sac. It can also be known as the embryo sac. See, I have given female gametophyte or embryo sac. This embryo sac, when it fuses and it forms zygote. Prior to that, how the female gametophyte was formed, here you can see there is only one embryo sac. Hence it is known as monosporic embryo sac, because only one embryo sac is being developed from the 4 megaspores. If you remember what I had taught earlier, from the 4 megaspores, only 3 degenerate, only 1 remains functional. That functional megaspore is again, it is forming the embryo sac. So it is from 1 megaspore. So it is known as monosporic embryo sac. If suppose 2 megaspores are involved, then it will be known as bisporic. Similarly, tetrasporic, till 4 tetrasporic. So mostly it is a monosporic embryo sac. It's a monosporic embryo sac. That is, it is developing from 1 megaspore. So we have seen the zygote is formed. We had seen endosperm formed. We had discussed also how and how many types of endosperms are there. Now we will see further development of zygote. How the further development of zygote takes place. So here I had explained just now how the zygote is formed. Here, okay. Zygote is formed here. Here endosperm and zygote is also here. This zygote. How this zygote will develop further? How the further development of zygote takes place? Zygote is now 2N, 1 N of the egg cell and 1 N of the male nuclei. Now it is 2N diploid structure, that is zygote. This zygote, we focus on this zygote. This zygote will divide into 2 cells. That is, one will be known as the terminal cell and another is known as the basal cell. It will divide transversely into a terminal cell and a basal cell. This terminal cell will again mitotically, it will divide into, vertically it will divide into 2 cells, and the basal cell will divide transversely into 2 cells. Okay. This is a 2-celled proembryo. Because embryo is not formed yet, this is the zygote. This is the 2-celled embryo. Now, 2-celled proembryo, before the formation of embryo, this is the 2-celled proembryo. Here you can see this can be called it as a 4-celled proembryo. This is a 4-celled proembryo. After that, this terminal cell, this is the terminal cell, this is the basal cell. This terminal cell will again, it will divide at right angles. Then it will form 4 cells. Here is the basal cell, 2 basal cells, 4 cells. Again, this 4 cells will divide and it will give rise to, mitotically, it will give rise to 8 cells. Anterior cell and posterior cell. That is the 8-celled structure. Okay. This 8-celled structure of the terminal cell, this 8-cell structure will again divide periclinally and it will give rise to 1, 2, 3, 4, 8 and 8 total. That will be a 16th cell. Now it has become a 16th celled. It had divided into 8 cells, then periclinally division will take place and it will give rise to 16 cell structure. Now the embryo at this stage, it's globular in structure. If you see, it is looking like a ball-like structure. It is globular in structure. Meanwhile, what happens in the basal cell, these 2 basal cells, it will again divide. The cell which is nearer to the embryo, it forms the hypophysis. It will form the hypophysis. And the structure just below the hypophysis, it will divide mitotically, it will divide. And it will form the 8-celled structure, 1, 2, 3, 4, 5, 6, 7, 8 and one large haustoria. This is the suspensor. At the embryo, at this stage, it is known as the globular embryo. Now it is known as a globular embryo. It has come into the 16 cell stage. Now it is no longer a pro-embryo. It will be called as a globular embryo. It is a 16 cell structure. So what you are seeing is the hypophysis, the suspensor. What is the function of the suspensor in this? What the suspensor will do, see, this is the ovule, okay, the nucellus region, this is the nucellus region. Here the endosperm. This 8 cell structure, when it is there, it has developed into an 8 cell structure, proembryo. So what is happening is, it is pushing itself inside the nucellus. This suspensor, it is pushing the embryo inside through the suspensor cell, it is pushing it inside the sac. Just to gain more nutrition from the endosperm, it is pushing it inside. And the thing, enlarged thing what you are seeing is the haustoria. What is the function of the haustoria is that it will develop more nutritive tissue from the adjoining areas of the nucellus. Suspensor is pushing the embryo towards the endosperm inside more to take nutrition. Other than that, if more nutrition is required, it is the haustoria which collects the nutrition, towards the from the adjoining cells, haustoria. So this is how the zygote will develop. It will go inside. Now after that, when it is gaining entry, we had seen it till the 16 cell stage, it will develop, divide, and now it has formed a heart-shaped structure. Embryo, it's heart-shaped. Here is the hypophysis. The suspensor. Now if you see, it's the heart-shaped structure. After that, this hypophysis, it will again divide. Hypophysis will divide again with the embryo. Even the embryo will go, more divisions will take place in the embryo. Now after that, it will look like a horseshoe-shaped structure. After heart-shaped structure, it will again divide, keep on dividing. These are the cells. It will keep on dividing, embryo will keep on dividing. Now it looks like a horseshoe-shaped structure. Here are the suspensors. Yes, you think the suspensor will be remain till now? The suspensor will also divide. The suspensor cells, these will also divide. That is, the hypophysis is helping in the division. When the hypophysis is helping in division, means what it will form? Hypophysis, when it is present anteriorly towards the embryo, hypophysis will help in the formation of epidermis, and the cells below the hypophysis, all the suspensor cells which will be there, they will help in the formation of root cap and radicle. Root cap and radicle will be formed from these cells. After some time, this is all this, what are these things are taking place? It is all taking place inside this. After some time, when the cotyledons are fully developed inside this only, when it has been fully developed, it will look somewhat like this. That it will have 2 cotyledons, then cotyledons, hypocotyle, plumule, from where cotyledons will develop, then radicle, and the root cap which will cover the radicle. The full development, all full development will take place here. Now the embryo is fully formed. It is a dicot embryo because 2 cotyledons are formed, and this is how formation of a dicot embryo takes place. Now we will see a monocot embryo. How does a monocot embryo takes place? We have seen in a dicot embryo, division is such, like it will go for the 4 cell stage, 2 cell stage, 4 cell stage, 16 cell stage, but this is not happening in case of monocot. In monocot, the zygote, it will develop into 3 cells: basal cell, middle cell, and terminal cell. In monocots, it is very simple. In monocots, It is the middle cell which will take part in the division, and it is the in monocot this middle cell is only forming the scutellum, plumule, hypocotyle, and it is a single cotyledon. In monocot it is single cotyledon. This is how it will take place in a monocot. It will basically divide from the middle cell. Terminal cell and basal cell do not take part in, do not have so much role in the formation of a monocot embryo. Now, when we have dealt with monocot embryo, dicot embryo, endosperm, so now we will see seed. What is seed? Can we say the fertilized ovule as seed? Fertilized ovule? Can we say it as seed? We had seen how the embryo develops, how the endosperm is formed, and what is the fertilized ovule? Fertilized ovule is your seed. Now, seed also, it is being divided into two, albuminous and non-albuminous. In non-albuminous, endosperm does not remain. Endosperm is used up. Non-albuminous, endosperm is used up completely. Like in peas, you have seen peas, endosperm is completely used up. In albuminous seeds, peas, groundnut, two examples. In albuminous seed, endosperm is present, like a rhiza, maize, castor, endosperm will remain. So based on that, seed is being divided into two, albuminous seeds and non-albuminous seeds. Then if you see endosperm, sometimes what is happening, perisperm is also persistent. Perisperm is also persistent in some plants. Perisperm is also there. It will remain persistent. What is perisperm? Remnants of the nucellus. Perisperm is remnants of nucellus. Why the seeds are so important? Why always we are emphasizing on the seeds? Seeds are carrying for the future plant. For the future plant, we need seeds. So these seeds carry hereditary material, these seeds carry storage, these seeds are having the future plant, these seeds can also be used for hybridization. So there are so much functions of seeds, so much uses of seeds, because seeds are the future plant. So we have to conserve seeds. So based on that, so there are so many ways, like one is apomixis. First, if we go for apomixis, what is amphimixis? Amphimixis means fertilization, that is the fusion of male and female gamete to form zygote, and further development will take place. Now what is apomixis? Apo means without mixing, that is no fusion of male and female gamete, that is apomixis. What is polyembryony? Polyembryony means poly, more than one. So development of more than one embryo. Development of more than one embryo is known as polyembryony. We had seen that a monosporic embryo sac means only one embryo will develop from that. But if it is polyembryony means all the 4 megaspores, of which earlier what it was said that the 3 megaspores degenerate on only one functional megaspore remains. But in polyembryony what happens? All the 4 megaspores can become functional. Some may be fertilized, some may not be fertilized, some directly develop from the means it is a haploid megaspore. It can also directly give rise to an embryo. It can give from the nucellus also embryo could be formed. From the integument also embryo can be formed. Means so many places embryos are being forming. So that means polyembryony, development of more than one embryo. This is polyembryony. Now we go for a quick again recapitulation like we started with. I started with the dicot embryo, but now I'll start with the monocot embryo. We go for a recapitulation. We see a zygote is formed in a monocot plant. Then the zygote goes for embryonal cell division, that is, it is an unequal division. After the unequal division, you see there are three cells are formed: the upper basal cell, the middle cell and lower embryonal cell. Now of the three cells, the middle cell is active. The middle cell divides and it is forming the globular embryo, as you can see in the figure. Now this globular embryo will divide and redivide and it forms the radicle and plumule, which gives rise to the radicle, plumule, hypocotyle and cotyledon. If you see, there's one cotyledon. Spelling of cotyledon is COTYLEDON, cotyledon. Okay. Now next, if we see the development of a dicot embryo, I went for the female gametophyte just to make you clear for the zygote, how the zygote is formed. I went for double fertilization again. As you can see from the diagram, the synergids are degenerating. What was the function of the synergids? They were rooting. They were providing the root to the male nuclei so that it fuses with the egg and it forms the zygote. You can see in the diagram, zygote 2N diploid structure. Another fuses with the secondary nucleus and it forms the primary endosperm nucleus, that is 3N. Earlier it was 2N, polar nuclei. Then antipodal cells also degenerates. Then the zygote, it forms into two-cell structure, that is, it is now known as the two-celled proembryo. Upper cell is known as the terminal cell, below is known as the basal cell. Then the zygote, again it will, I had explained it in that how it will go for a 4-celled proembryo. Then the 16-cell structure, it formed the globular embryo. Then just below to the globular embryo will be your hypophysis, and then the topmost is the suspensor cell, which will push the suspensor. It will push 8-cell structure. It will push the embryo, developing embryo towards the endosperm so that it gains maximum nutrition from it. Then again, when it divides and divides, it forms a heart-shaped embryo. Then it divides again to form the radicle, plumule and the two cotyledons. Now, we went for the fruits. What are fruits? There are two types of fruits, that is true fruits and false fruits. True fruits are fruit which will develop from the ovary, and false fruit develop from other parts of the ovary. If it develops from the ovary, example tomato, grapes, banana, false fruits. Other than the ovary, like thalamus, apple, pears. Now you can see true fruits like in mango, coconut, epicarp, mesocarp, fleshy part mesocarp, the innermost endocarp. Okay. Then in the false fruits, thalamus, which develops from albumina. Now seeds we had seen, why seeds are there? The seeds have divided into two, albuminous seeds and non-albuminous seeds. Albuminous seeds have residual endosperm and non-albuminous seeds do not have residual endosperm because they are consumed by the growing embryo. Albuminous seeds, example wheat, castor, maize. Non-albuminous pea, groundnut and beans. See the example of the albuminous seeds, the diagram is clearly given. Non-albuminous seeds. Now, what is apomixis? Seeds produced without the union of sperm and egg, and it occurs naturally in about 400 plant species. Apomixis can be triggered because at low temperature also it changes to plant opts for apomixis rather than true fertilization, amphimixis. Low temperature, or we can go for auxins and gibberellins, the plant hormones, they can trigger apomixis. Polyembryony, more than one embryo during the development of the seed. We have seen polyembryony. So we have completely seen the chapter, and this, I think you have understood the chapter reproduction in flowering plants.